Namaste. Welcome to biology class. In the chapter Biomolecule, we have been learning about hexose sugars in the previous video. Today, we will be learning about the pentose sugars and also disaccharides. We know that the pentose sugars are made up of 5 carbon atoms and its formula is C5H10O5. We also have learned that the examples of pentose sugars are ribose, deoxyribose, arabinose and so on. Ribose and deoxyribose, they are essential for the formation of nucleic acids, that is, which form the genetic material in most of the living organisms. Ribose plays an important role in the RNA and deoxyribose plays an important role in DNA. Now, let us know the structure of these pentose sugars. Coming to the ribose. Ribose, we have the ring which is made up of 4 carbon atoms and 1 oxygen. So, it is called as a furanose ring and it has got 5 sides, so it is called as a pentagonal ring also. Now, here this is a ribose structure. The deoxyribose is also having a same structure with a pentagonal ring and the furanose ring. But the difference is at the second carbon atom. Yes? Here in the second carbon atom, you can see that there are OH and H and you have OH and H over here in the second and third. Here you see in the second carbon atom, 1 oxygen is less. That is the reason why we call this as the deoxyribose. Deoxy means from ribose, 1 oxygen is less. That is why this is the main difference in ribose and deoxyribose. Other than that, we also have few differences between the ribose and deoxyribose. The ribose has got 2 functional groups which makes it more reactive. Due to the lack of oxygen, there is only 1 functional group in deoxyribose and because of which it is less reactive. Remember, more the number of functional groups, more reactive it is. And so, this ribose, as it is more reactive, it is less stable. Whereas, the deoxyribose, as it is less reactive, it is more stable. That is the main reason probably why DNA is the genetic material in most of the organisms when compared to RNA as the genetic material. So, this is about the pentose sugars. Now, let us learn about the disaccharides. Now, as the word suggests, disaccharides. So, it is made up of 2 monosaccharides. These monosaccharides are joined by a glycosidic bond. Now, let us see few examples of disaccharides. The first one is maltose. It is also called as the malt sugar. This maltose is actually made up of 2 monosaccharides or rather 2 molecules of glucose. So, you can see the glucose structure over here. Glucose has got a pyranose ring, hexagonal ring. Yes, we have been learning it in the last video. Now here, let us count the number of carbon atoms. 1, 2, 3, 4, 5 and 6. Yes. Now coming to the next glucose molecule, you have 1, 2, 3, 4 and 5. Yes. So see here, the bond is between carbon atom 1 and carbon atom 4. Yes. So that is why this glycosidic bond is called as 1,4 glycosidic bond. Understood? So, the bond name is given based on the number of carbon atom it is actually sharing the bond with. Yes? Okay. So, this is about the malt sugar. Now, in this we already know that glucose has got aldehyde group at the terminal end, which is involved in the formation of the bond over here. But the other glucose molecule is not sharing the aldehyde group. So, its functional group is intact. That is the reason why maltose is a reducing sugar. So, maltose is a reducing sugar as one aldehyde group is already intact. That is why it is called as a reducing sugar. Now, coming to the next one, that is sucrose. Sucrose is mostly seen in sugarcane. That is why it is also called as the cane sugar. This sucrose or the disaccharide is actually formed by glucose and fructose. Glucose has got pyranose ring and fructose has got the furanose ring. Now, here you can see it very clearly. This is the pyranose ring of glucose and this is the furanose ring of fructose. Here you can see that the bond is formed between the first carbon atom and the second carbon atom. So, first has got the aldehyde group, which is a functional group for glucose, and second one has got the ketone group, which is a functional group for fructose. As both the functional groups have been involved in the formation of this 1,2 glycosidic bond, there are no free functional groups. So, the sucrose molecule is called as a non-reducing sugar. Now, coming to the last one, that is lactose. So, lactose is a disaccharide which is made up of a glucose molecule and a galactose molecule. Both of them have got aldehyde group as the functional group, but it forms a 1,4 glycosidic bond wherein the aldehyde group of galactose will be free. That is the reason why lactose is called as a reducing sugar. So, this is the concept of disaccharides. Now, most of you might have been wondering that why there are separate bonds over here. I just wanted to show you how these sugar molecules are involved in the formation of nucleic acids. Now, in the first carbon atom, there is one nitrogenous base which gets attached to this, and at the fifth carbon atom, there is a phosphoric acid which gets attached to this. Then, this ribose sugar becomes RNA, that is nothing but ribonucleic acid. It becomes ribonucleic acid. In the same way, for the deoxyribose, if at the first carbon atom the nitrogenous base attaches and at the fifth carbon atom the phosphoric acid attaches, then this becomes DNA, that is nothing but deoxyribonucleic acid. So, this is how the RNA is formed based on the ribose, and deoxyribose is involved in the formation of DNA. So, this is about the pentose sugars and disaccharides and how these pentose sugars are involved in the formation of nucleic acids. Hope the concept is clear for you. Thank you.